So I'm Naveen Kumar, and I'm European project manager for Irish Bioeconomy Foundation based in Tipperary, Ireland. We deal with a few European projects like Copilot, Primed, Rural Bio-op, Approvals, Bioperist, to name some. And along with European project, we also own a pilot demo facility in Republic of Ireland, Tipperary National Bioeconomy Campus. So today I want to talk about a project Rural Bio-op, in which the initiative is to increase the knowledge awareness to the community, providing the knowledge on the business model and the technologies. And here I would like you to talk on the technologies you are using in your research. So please feel free to introduce yourself and you can start the slide. So I'm going to switch off my cam and let you speak. Thank you.
Thank you, Naveen and Kevin, for this opportunity. So I am Pooja. I'm a PhD researcher based at University of Galway. I'm working in the School of Biological Sciences with Vincent O'Flaherty's group here in Galway. And previously, I have worked as a research fellow in India for 3 years with reputed government institutes in India. And today I'm going to talk about one of the most emerging topics in Ireland. And I'm going to share my screen now. So the topic for my today's discussion is the green gold. I'll be talking about valorizing the residual biomass through the anaerobic digestion technology in a green biorefinery concept. To begin with, I would like to introduce my project. So my research is a part of a big project called U-Protein that's unlocking the protein resource opportunity to evolve Ireland's nutrition. In this project, what we are aiming towards is to re-engineer Ireland's agroecological system to look for different protein crops that could be produced locally. And we are involved in the task 4 of this project, where we are trying to biorefine or biotransform the residues that are being generated after protein extraction to value-added products, be it volatile fatty acids, biofuels, fertilizer, and other functional materials. Going ahead, I would like to first explain what a biorefinery is. A biorefinery is similar to a petroleum refinery, where a biomass is processed in a sustainable manner to a spectrum of value-added products and energy. So particular components of a biomass, be it proteins, sugar, lignin, oil, or fats, are processed via a technology to food, feed, chemicals, and materials. And this processing aligns with the Sustainable Development Goals of the United Nations. Now, what is the need of this biorefinery? So in the conventional times, what has been happening was right from the beginning of the harvest of the crop to the production of the value-added products, there were certain residues being generated throughout this processing. And these residues were left underutilized and were considered as waste. But with the advancement in technology and using the new technologies from the beginning of the harvest of the crop to the production of the value-added products, in a circular bioeconomy approach, whatever is being produced at every step of the process can be utilized or upcycled to value addition. Now, I would like to explain through this through an example. So here you can see a crop has been harvested and is being processed. So while from the harvest to the processing of the crop, there are certain side streams being generated. And these agriculture side streams could be rich in protein or as well in cellulose or hemicellulose-rich components. So the protein-rich substrates could be targeted for food ingredients, and they could be concentrated for food industry. However, the cellulose and hemicellular-rich substrates, which has high sugar content, could be fermented to produce ethanol or other value-added products, such as volatile fatty acids. So this is how different streams of a crop could be subjected to different fermentation processes or different value additions. Now, going ahead on a broader context, I would talk about the green biorefineries, that how the previous circular system could be put into a practical aspect. As we know, Ireland is an agriculture-based industry, and even after meeting the meat and poultry requirements, we have surplus amount of grass left. So that grass could be subjected for protein extraction, and the residues that has been generated while extracting the proteins could be upcycled to other value-added products. Now, what is the need of this grass to be used for protein extraction? As more than 80% of the protein in the European Union is being imported in the form of soybean meal. And these imports not only put pressure on the logistics, causing environmental pollution, but also affects the economic growth of the country. So in this process, we are subjecting the grass through the screw press for protein extraction, rich green juice, and while we are extracting the juice, there are certain residues left behind. On the other hand of this process, a residual cake is being generated, which is rich in fibers, and we are subjecting that cake to anaerobic digestion process. Anaerobic digestion is a well-known technology, which I'll be talking about in my next slide. And through this anaerobic digestion process, we are generating multiple products such as digestate, which could be sent back to the soil for nutrient recovery, volatile fatty acids, which could act as platform chemicals for further use in food, pharmaceutical, or chemical industry, and the generation of the biogas, which could be used as an energy source. So it will be a zero waste technology for a circular economy approach. That means every blade of the grass has taken into account. Now, on a practical aspect, I'll discuss more about the anaerobic digestion technology. It is a microbial marvel. So this technology is well established and is well known for energy recovery from the biomass. Right from the beginning of the anaerobic digestion process, the first step of this process is hydrolysis. And in the hydrolysis, we have certain organisms, we call them hydrolytic organisms, which secrete certain extracellular enzymes to convert the complex organic matter, be it lignocellulosic, carbohydrates, proteins, fat, into simple monomers. And these simple monomers act as a substrate for the next process. So it is a sequential process in which the substrate, the product of the one process actually acts as a substrate for the next process. So after the hydrolysis, we have acetogenesis involved. In the acetogenesis, we have certain fermentative bacteria, which further degrades the monomers of this process to produce alcohols, aldehydes, and volatile fatty acids. And these volatile fatty acids are the intermediates of this process, which could be used as platform chemicals. Going ahead, while the fermentation of nitrogenous compounds like amino acids and carbohydrates usually occurs in the acetogenesis, this step is strictly carried out by anaerobic bacteria and archaea. Acetogenesis involves the metabolism of acetogenesis products to acetic acid, hydrogen, and carbon dioxide. Coming to the final stage of this process, that is called methanogenesis, which results in the production of biogas, and the main component of this biogas is methane. The technology is well known for its energy recovery from the biomass. However, the resources that are recovered from this anaerobic digestion processes are biogas, which could be used as a biofuel, the digestate, which could be used as a fertilizer for the soil, and as well as the carboxylates such as volatile fatty acids, which could be further used as platform chemicals for further utilization. Going ahead, what we are doing in this project is, so we are trying different feedstock, be it grass, mash, or oca, faba beans, for the protein extraction through a process called twin screw extrusion. It is a mechanical process in which the proteins are being extracted. And what we are particularly focused on is after the protein extraction, the residues that are being generated. So as you can see on the right-hand side of the slide, the residues which are being generated, we are subjecting it to anaerobic digestion process. And through controlling certain physical parameters, we are trying to produce volatile fatty acids. My research is particularly focused on butyric acid production, and it holds certain applications in pharmaceutical and food industry. And it is used in the flavoring purposes as well. And once these acids are being produced, the residue is subjected for biogas production, which is another product of this process. And once the biogas has been produced, the digestate that is left behind can be recirculated back to the soil for the nutrient recovery. So this is how we are closing the loop of this circle. And this is a practical application of how we are doing it. So this is a lab-based study that we are doing. There are 3 reactors you can see. These are called leach pet reactors, and adjacent to them you can see blue LED screens. So these are the controllers of these reactors, and the pH is being monitored through these. These reactors are double jacketed to control the temperatures and several process parameters that we are trying to uh, increase the efficiency of butyric acid production in these processes. Going ahead, if these processes are successful, it will help in reducing the feed imports in Ireland. That will eventually we'll go for something local, and the GHG emissions which are related to the logistics involved in the imports will also be reduced. And third, it is a circular economy approach, so all the residues that are being generated in the process are being upcycled, technically leaving no waste. And fourth, it will lead to some business opportunities inside for the local as well as in the international market. Thank you for listening.
So thank you very much, Pooja, for joining this webinar and giving a lecture. It was very self-explanatory and very informative. So I would like to thank you on the behalf of IBF team and Kevin Ryan, our senior European manager, and Rural Bio team. So thank you very much. And I don't particularly have any questions, so it was really good. So thank you to the University of Galway and Vincent O'Flaherty's team.